Biological membranes, also called plasma or cell membranes, form barriers to provide specific environments for their contents. Membranes act as selective barriers, as they restrict the movement of some substances while allowing the movement of others. Plasma membranes are found surrounding all cells, differentiating the internal environment of the cell from the external or extracellular environment. In addition, they are found surrounding structures within eukaryotic cells, such as the nucleus, and membrane-bound structures called organelles that have specific functions within the cell. The fluid Mosaic model describes the composition and structure of plasma membranes. The fluid portion is composed of a dynamic phospholipid bilayer, and the Mosaic portion refers to the presence of not only phospholipids, but proteins and other types of lipids found within the membrane. If you will recall, the head of a phospholipid molecule contains a phosphate, glycerol, and a polar or charged group, making it hydrophilic. Attached to the head are two nonpolar fatty acid tails. As in fats, these fatty acids can be saturated or unsaturated. So if we drop some phospholipids in water, the hydrophilic heads interact with water molecules, while the hydrophobic tails interact with one another to exclude the water molecules. These spherical droplets are called micelles. But phospholipids in aqueous solutions can also form more complex arrangements called bilayers, with two rows of phospholipids in a spherical shape. The hydrophilic heads interact with the exterior or the interior of the bilayer sphere, while the hydrophobic tails interact with each other. If we take a membrane surrounding the entire cell, the outer layer would be towards the extracellular fluid, while the inner layer would be towards the cytosol, which is the solution we find within cells. Now, the fluidity of the plasma membrane depends on many factors. Membranes are considered fluid to at least some extent because the phospholipid molecules move. They rarely move by flipping from one layer to another. Why do you think this would not be a common occurrence? Well, the hydrophilic head would have to travel through the hydrophobic tail region to get to a different layer. That's just not natural. So the phospholipids like to remain in their original layers, but they move at an extremely fast rate laterally. That means the phospholipids move around in the same plane or layer. That way the hydrophilic heads are interacting with each other, and the hydrophobic tails are interacting with each other. Perfectly natural. In addition, the shape of the phospholipids' fatty acid tails has a lot to do with fluidity. If the fatty acids are unsaturated, meaning they have at least one double bond between the carbons of the skeleton, then they will kink. This kink keeps the phospholipid molecules separated from one another, increasing the fluidity. If the tails are saturated and have no double bonds, they are relatively straight. This allows the phospholipid molecules to pack together closely, decreasing fluidity as the molecules cannot move around as much. Substances that are not very fluid are called viscous. And other molecules embedded between the phospholipids can affect fluidity. Esters, such as the phytoesters found in plants and the cholesterol found in animals, tend to act as buffers to maintain a certain membrane fluidity. If the temperature decreases, the phospholipids would move around more slowly and compact closely together. As a result, fluidity would decrease. The esters, such as these cholesterol molecules, interact with the hydrophobic tails and keep them from compacting too much. This maintains fluidity. Also embedded in the sea of phospholipids are proteins. This diagram shows different types of membrane proteins. If you compare the exterior side facing the extracellular fluid to the interior side facing the inside of the cell, they are very different. This is because the interior proteins have a different job to do compared to the exterior proteins. Integral proteins have both hydrophobic and hydrophilic regions, as they span the width of the membrane. Many integral proteins act as passageways to allow substances in and out of the cell. Peripheral proteins are not embedded in the lipid bilayer. They are bound to the surface of the membrane or to an integral protein. Many peripheral proteins have enzymatic functions. So how do substances move in and out of the cell, if cells are protected by this dynamic membrane. First, let's review a cell's environment. Cells contain and are bathed in an aqueous solution, which consists of a solute and a solvent. When we place a solute like sugar into a solvent like water, the polar water molecules interact with the polar sugar molecules, and the sugar dissolves in the water. But the solute doesn't stay in one place. The energetic molecules randomly move around and bounce into one another. Eventually, the solute molecules are as far away from one another as possible. And this is diffusion. All atoms and molecules have an inherent energy which allows them to move from an area of higher concentration to an area of lower concentration. Does diffusion occur in a cell? Well, let's look at this simulation. Here we have a selective membrane. The small red particles can move across the membrane. There are many red molecules on one side of the membrane, so we have a concentration gradient. There is a high concentration of solute on one side and a low concentration of solute on the other side. Based on the concentration gradient, will the red molecules cross the membrane? And if so, how many will cross? As you can see, about half of the solute molecules cross the membrane by diffusion. Now each molecule is as far away from the others as it can be, and we have reached an equilibrium. But remember, molecules have an inherent energy, so they are constantly moving around. And we call this a dynamic equilibrium. But there are tons of different molecules and ions. Cells cannot allow everything to enter. So let's consider a semi-permeable membrane. Some substances are allowed to diffuse across, while others cannot. Here's a really big molecule. Do you think it can fit through the pores of the membrane? Nope, too big. What about this charged molecule? Can it travel through the hydrophobic fatty acid tails of the bilayer? Nope, too charged. What about this molecule? Water. It's polar, but it's really, really small. Just right. The water molecule was small enough and only had partial charges, so it could get through the membrane. We give a special name for the diffusion of water across a semi-permeable membrane: osmosis. Don't think that osmosis is different from diffusion. Diffusion is a general term that we use for any substance moving from a higher to lower concentration. Osmosis is specifically the diffusion of water across a membrane. But what about cells? What substances are selected to cross the plasma membrane? Some molecules can quickly cross the plasma membrane by directly diffusing across it to reach an area of lower concentration. These molecules are typically very small and nonpolar, as they need to make their way through those hydrophobic tails of the phospholipids. This is simple diffusion, so it is a passive process. No energy is used. So extremely small and nonpolar molecules like oxygen and carbon dioxide diffuse very rapidly across the plasma membrane. More complex but still small and nonpolar steroids and fat-soluble vitamins rapidly diffuse across the plasma membrane. But what about water? It's definitely small, but it is polar. So while it does diffuse across the plasma membrane, it diffuses at a relatively slow rate. And there are many, many water molecules in the cytosol and extracellular fluid, so they can diffuse more readily than many other molecules because of their vast numbers. But water still doesn't directly diffuse across the membrane fast enough to accommodate the needs of most cells. This is accomplished by our next type of diffusion. Facilitated diffusion is when a protein assists in the diffusion of a substance across the membrane. The cell may require these substances, but the molecules are just too large and are too charged to cross directly. Again, this is still a passive movement, as the protein is just helping the molecules diffuse. So while water can slowly diffuse directly across the membrane, it mainly diffuses with the help of a channel protein called aquaporin. It is called a channel protein because there is a hole that runs through the protein to allow water to flow from one side of the membrane to the other. Much of the exterior of the protein is hydrophobic, as it must interact with the hydrophobic tails of the phospholipids. But the interior is hydrophilic, as this portion interacts with the water molecules, allowing them to diffuse into the cell at a much higher rate compared to diffusion directly across the membrane. Ions like this potassium cation do not directly diffuse across the plasma membrane and must utilize channel proteins called ion channels. As ions are charged, they not only move due to a concentration gradient, but also an electrochemical gradient. In fact, most ion channels are gates and will not open unless the outside has a particular charge in comparison to the interior. If the exterior of the cell has an overall positive charge in comparison to the interior, the cation will most likely move in the direction of that negative charge. The plasma membrane is somewhat permeable to glucose, as it is a small molecule, but it is still too big and too polar to diffuse at the rate required by most cells. The GLUT1 protein is a carrier protein in which glucose binds to the exterior side of the protein, which induces a conformational change. This means the protein changes shape, opening a channel that the glucose molecule can use to diffuse into the cell. Now that we know how substances can diffuse across membranes, let's explore osmosis in more detail. You know it is the movement of water across membranes, and most osmosis occurs through aquaporins. But will the water flow into the cell or out of it? That depends. Tonicity is the comparison of solutions to determine the direction of water flow across a membrane. Tonicity is relative, as we must be comparing two solutions. For example, the solute concentration of a solution inside a cell to the solute concentration of a solution outside a cell. We are assuming that the solute cannot diffuse, so only the water can flow across the membrane. The three types of tonicity are isotonic, hypotonic, and hypertonic. Remember, we have to be comparing two solutions to use these terms. We will examine the three different types of tonicity by using a red blood cell. We are assuming the membrane is not permeable to the solutes we are investigating, so we are only looking at osmosis, the diffusion of water. Isotonic solutions have equal solute concentrations. If a red blood cell is placed in an isotonic solution, there is a dynamic equilibrium. Water molecules still move in and out of the cell, but there is no net movement of water, meaning the same amount of water that moves out will move in. In isotonic solutions, the solute concentration outside the cell is the same as the solute concentration inside the cell. This is a red blood cell in an isotonic solution. It normally has that depression in the center. We say that the red blood cell is in osmotic equilibrium in isotonic solutions. A hypertonic solution has a greater solute concentration when compared to another solution. In this case, there would be a greater net movement of water out of the cell. This is because only the water can diffuse, not the solutes. When a cell is in a hypertonic solution, there is a greater concentration of water inside the cell. As there is more water inside the cell, it moves outside the cell by simple diffusion. Here's a good way to remember it. Water always moves to the area of greater solute concentration. So the red blood cell in a hypertonic solution would lose water and crenate or shrivel. A hypotonic solution has a lower solute concentration compared to another solution. If a red blood cell is placed into a hypotonic solution, there will be a net movement of water into the cell. As there is a higher water concentration outside the cell, the water moves into the cell by simple diffusion. Again, the water is moving to an area of higher solute concentration. A red blood cell in a hypotonic solution will swell and potentially burst if it takes in too much water.